for a few more minutes for others to join. We'll start at 7:05.
Should we begin?
To talk more on this topic, we have our guest speaker, Shivam Chowdhury sir. He holds an MSc Cognitive Science in Brain Computing and Interface and Human Robot Interaction. Without much delay, let me hand over to you, sir.
Thank you, Ashwarya. So I think before beginning this talk, I want a small, demographic information. So everyone who has some idea about neuroscience, please raise your hands. Oh, my voice is breaking. Is it better now? I didn't change anything, but right. Okay, I think we have quite a bit of people. Okay, and people who have computer science or engineering, electrical engineering backgrounds. Okay, then. So I'll share my screen now, and we'll begin with the talk. Okay, can you see my slides? I okay. Can anyone speak up because I won't be able to see the chat box.
Yes, the slides are visible, sir.
Okay, so welcome to today's talk on introduction to brain-computer interfaces. So before we begin, I would like to show you a video. So have a look. So what is happening here is that the person who's sitting on the chair, the woman, is paralyzed and cannot move her limbs at all. And the technician in the back has installed a invasive brain-computer interface into her cortex, into the brain, and she is able to move this robotic arm by just imagining the movement. Right. So I think this is one of the reasons why we are interested in brain-computer interfaces. And please stop me if you have any questions. Type it in the chat box or just speak up. Okay. All right. So these are the things that I will be sharing about in the coming slides. This is just for my reference. So, okay. So before I get into brain-computer interfaces, I would like to tell you some things about neuroscience. So on if you see the left picture, it is the human brain. Our human brain is divided into four major lobes: the frontal lobe, which is the front side, the parietal lobe, which is right over here. If you can see, if you see me also, and the back of our head is the occipital lobe, and on the sides, both the sides, we have the temporal lobe. And in the picture, if you see my laser pointer, this is the brainstem, and connecting to the brain, the brainstem connects the spinal cord and the rest of your brain, and on the back of the head you have the cerebellum. So the entire lobe, these four structures are also called cerebrum or the cortex. So if I say cortex, it means these four structures. So this is here is a picture of a neuron. So a neuron is a functional, is the smallest unit of a human brain. What a neuron does is it collects information from all other neurons from this part of the body of the neuron. These are called the dendrites. It takes the information, computes whatever it has to, and then sends an output signal through the axon. And from these terminals, these are called axon terminals. It dissipates that information. It releases certain chemicals and dissipates that information to other neurons. So the information is sent as an electrical impulse through this axon, and the electrical impulse causes a release of chemicals. So this is called electrochemical reaction. This is called electrochemical signal processing. So how do neurons communicate? As I mentioned in the previous slide, uh, these are the electrochemical signals, and they are called action potentials. If you see the picture here, this is a typical action potential. It is also called as a spike. The sensory input from our sensory receptors, be it the ears, the eyes, or touch or heat sensors, send information to the brain using via neurons using action potentials. The brain takes that information, integrates it with the information that it has already stored, and then sends out signals to our through the motor neurons in the form of, again, action potentials, and that signal stimulates our muscles so that we see the movements that we create in the everyday. So, action potential work on the principle of one or none. So, if it fires, then it sends a signal. How different signals are differentiated is via the frequency of firing. So, if I slightly touch my palm, it will send a low frequency signal to the brain. If I touch it harder, then it will send a high frequency signal. So this is what a typical EEG looks like. EEG stands for electroencephalography, and it is recorded by placing electrodes on the surface of your head. All right, this is a 64 channel EEG headset. So now that you are familiar with basic neuroscience, let's go for some machine learning. These slides won't make you an expert, but in traditional programming, we see, I see a hand raised. Grishma, yes. Okay, never mind. Let's move. In traditional programming, you provide the data and you provide the rules and you get the answer. Like y is equal to mx plus c. M and c, so y is equal to mx plus c is equation of a line, right? So x and y are the two variables. Y is the answer. X is the value that x is the point whose corresponding y value we want, and m and c are the rules how we compute the y coordinate of the x coordinate on a straight line. But something different happens in machine learning. Okay, so we have a question. If a person is doing a particular task, will the brain produce same EEG graph? Yes, definitely. So if I am shown if I am shown a white cross on the middle in the middle of the screen, and watching that will produce same EEG response every time, but you won't be there'll be some random noise in the EEG signal that will hide that same signal. So there's a whole signal processing you have to do and when you do that, you will see that the EEG response is the same. There's a lot of noise that you have to go through, you have to wade through, but it's the same. Okay, did that answer your question? I will take that as a yes. Moving on. So, what machine learning does different is that we give the question and we give the answer to the machine learning algorithm. It takes out what rules it figures out the rules that we use to get those answers. So that is in EEG, what we do is we give the data, we give the EEG data, and we give the corresponding activity. Say the 30, okay, so half a second of data. I'm told to open my hand. So half second of data of EEG and hand opening is given to the machine learning algorithm. Multiple trials of that, and the algorithm figures out a rule on how on what is the exact signal in the EEG that corresponds to opening of the hand. Uh, Basically, yeah, we can. So the question is, can you identify a mental illness and neurological illness with EEG graph? Yes, yes. I mean, there's more to it, but the safe answer is yes, you can do that. But you need to compare it with someone, because EEG data, as we see here, in itself is not very informative. Granted, we know that these are eye blink signals, but from the raw signal you can't figure out much. So you need to see a normal EEG signal, and then you have to compare it with signals of person with mental illness or any neurological illness. Okay. So machine learning in brain computer interfaces. So there are two phases to it: the training phase, and in the training phase, we have the machine learning model or deep learning. We give the data, we give the brain data, and we give the corresponding labels. As I said, I give the half a second of EEG data to the model, and I tell it that it is hand opening. I also give it the data of hand closing so that it can differentiate, because you need to compare it with something. You cannot absolutely figure out what's going on, because the raw data is very convoluted. So the model will give out the rules with which what it learned, and how to differentiate between the two labels. And in the testing phase, you just provide the data with the new rules. It will tell you whether you have opened or closed your hands. Okay. So any questions on these two slides? You can type as well. I'm monitoring the chat box from another device, or you can speak up. I think that's a question that I can't answer. It's Revit. Okay, if we let's move on then. So we move on to, with that basic knowledge of neuroscience and machine learning, let's move on to brain-computer interfaces. Okay. So what is a brain-computer interface? We know that, as I mentioned earlier, our brains, if I want to move a hand, there's a lot of processes going inside my cortex, which will be the motor cortex right over here in the center of your head. And that those signals will go through my spinal cord, through my brainstem, through my spinal cord, through my muscles, and finally to the hand. But what about people who have a spinal cord injury, or people uh, who have a spinal cord injury, or people whose muscles have degenerated, or people with paralysis? So how will they won't be able to do it basically. So brain computer interface bypass the conventional mode of communication and provide a direct control between the human brain and the external devices. Yes, Neuralink also does brain computer interfaces. I think I answered that. I answered your question. Yes. So we bypass the conventional mode. We do not, because we have, suppose I have a spinal cord injury, the communication sent by the command sent by the cortex won't be able to reach my hand. So we bypass that. If you see the picture over here, electrodes are directly implanted to the person's head, and those signals are read by a computer, and they are decoded to give what the person is thinking. So this person who is paralyzed these are the implanted electrodes. This person is writing hello, imagining to write hello, and the computer has decoded using machine learning or deep learning algorithm that this person wants to say hello. Right. So there are many more applications of BCI, say neuroprosthesis. So if these prostheses could be used by amputees, people who have lost their limbs, people with muscle degeneration, or people with spinal cord injuries. They are not able to properly use their hands or limbs in general. So neuroprosthesis helps them regain some autonomy. Assistive devices, you can drive a wheelchair just by your brain signals. Communication, as in the previous slide we saw, person wanted to write hello and he imagined doing that, and we can see that on the computer screen. Assistive devices also include exoskeleton. Exoskeletons would also come under enhancement devices. Uh, some of the work that we are doing is on exoskeletons, but we haven't integrated with brain computer interface as of now. We are planning to do that. And rehabilitation. So when a person has stroke, they have very good they have some chance of regaining part of the control that they have lost in the stroke, but that needs to be done. That is, there is a critical period of about 2 weeks, 2 weeks to 2 months, within which they have to have a physiotherapist to help them regain some control. But with so much population and the dearth of physiotherapists, rehabilitation could happen with a robotic feedback as well. So a person imagines doing something, and the exoskeleton or the robot moves that part of the limb. By this, the axons, the neurons in the limb, are forced to make connections, are forced to remake the severed connections. All right. So these
Can
are
we make an
some.
extra limb with the help of? Can we make an extra limb using
Yes,
this
definitely.
mechanism?
But you will have some difficulty, because you have limited capacity in the brain.
But actually,
How many? Yeah,
human brain is only human brain is designed to manage only four limbs. But we need
right.
to generate an EEG graph for the extra limb. But it is impossible to generate EEG graph for managing the extra limb. But how will we be able to do that?
So, I mean, when our genetic predisposition is that we have two hands and two legs. We as Homo sapiens have two hands and two legs, right? So, to incorporate a third hand, we do not. So we can always control it, but not with the dexterity that I can control my fingers right now. So I have to do imagine movements. I have to imagine movements, but not do it with my hand. So you won't be able to use all three of your limbs simultaneously unless you train your brain. So that's called neuroplasticity. Am I clear? Did I answer your question?
Yes, but we need to design our brain and train our brain to do that a further task.
Yes. 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 Okay. So yeah. So I'll be talking about these three VCI techniques. So EEG stands for electroencephalography. The graph that I showed you earlier, that's an EEG graph. So that records brain waves from directly from your scalp. Okay, so I place EEG caps right on the scalp. I don't have to do anything else. ECOG is electrocorticography, in which you cut open the skull and you place micro electrodes on the surface of the cortex. You remember the cortex, the large portion of the brain. So you place it over there, and this is an invasive technique. And the third is electromyography recording, and these are recordings from directly on your muscles. So you place EMG electrodes on your skin, and they record your muscle activity. So if I keep a EMG on my bicep, and if I flex, where I will see a huge activity. I'll see huge amplitude in my EEG, EMG. There are more like fMRI, PET, FNIRS, functional near infrared spectroscopy, but these are the most common ones. This is a picture from one of the EEG caps that we have in our lab. So, as I mentioned earlier, the EEG caps record brain activity from the surface of the scalp. I mean, you can also record it if, even if the hair comes in between, it doesn't affect the recording much because the signals are eventually amplified. It is it is used very widely. It is relatively cheap. It has high temporal resolution, meaning you can get activity up to a millisecond. Differentiate between activity even if they are milliseconds apart. It is very precise, meaning see the cap is covering the entire brain, meaning we can make inferences about the brain activity. So if I see a huge spike or some activity on this electrode number 21, then I can say that that activity is related to the cortex that lies under that electrode. Okay, so electrocorticography. Uh, so electrocorticography records activity from the surface of the cortex. Like you have to cut open the skull, and then you place electrodes on the surface of the cortex. These recordings are very precise because there's no attenuation, and you get recording directly from the neurons that are present over there. So we place it wherever we want to monitor. We have to surgically open your head, and these are typically done on monkeys. They are done on monkeys because they share similar brain structure. I said similar, not exactly same, because we have similar visual cortex. Visual cortex is a huge covers a huge part of our brain. It has similarities across functions. Um, and we also do electrocorticography on humans. We place corticography on humans, but that can only be done if the person is already undergoing surgery for epilepsy, or they are there to get their brain tumor removed. Okay, so questions are being answered. Other than showing mechanical feats, can we help person share his thoughts or any mind reading applications? Yes, I mean there are EEG devices like Neophony or Muse that tell you the level of attention or meditation, or whether you're meditating or not. Or it also records your eye blinks. EMG. Let's move to EMG. So see the white. These are single-use very cheap, resolution. Temporal resolution meaning that it can record activity even even a millisecond, and it can also tell you precisely the location of the activity. So if you want, I'll share the PPT with you all. If you want, you can you can look at the videos from Backyard Brains where they open and close a hand using their EMG signals. It's very interesting. If you want to get started in BCI and have limited budget, then this will be helpful. So BCI is very vast. As I said, um, I told you about EEG, I told you about ECOG, I told you about EMG as well. But there are other also magnetoencephalography, positron emission tomography, functional near infrared spectroscopy. I think there's a typo, fMRI. There are a lot of recording devices, but EEG is most widely used, so I will be focusing on that predominantly. So, okay, so we have EEG headset and we want to do brain-computer interface. So how will we do that? Like, what's the next step? We can't just place the electrode and person and expect the person to just control an external device, right? So you have to have some kind of so you have to place the person in an experiment, not exactly an experiment. You have to tell the person to do something, or you perturb, or you show a person something, and based on their responses, you do something. So I'll so these are the three paradigms: P300, motor imagery, and steady state evoke potential. I'll be talking in detail about them. So how P300 works is that P300 gives a positive, huge positive peak. See here, P3 or P300 gives a huge positive peak starting from 300 millisecond after I show them a stimulus. So if I tell you to sit in a chair and I show you a letter A, that is your stimuli. I show you letter A on a monitor screen. So 300 to 350, it's now 350 to 600 milliseconds. It was originally found at 300 milliseconds, but it ranges from 350 to 600 as well. So the P300, you will see a huge spike in P300 wave. That response is mostly present at PZ, which is the parietal central area around this part of your brain. Um, Then we have the motor imagery or execution, which means that this is the activity in your motor cortex, the center of your brain, which is associated which lights up when you move a muscle voluntarily. So if I am doing any movement, my cortex is controlling all of that. My motor cortex is controlling that. So I record from the motor cortex to figure out what action the person is making. And there is the steady state evoke potential. It predominantly has two types of evoke potentials. You can evoke those potential in two ways: using images, that will be steady state visually evoke potential, or using tones or music, that is the steady state auditory evoke potential. So what happens here is that when I show repeated stimuli, this will elicit an EEG response. This will create an EEG response, and your brain signals will synchronize with the frequency of the stimuli. Okay, so I know there's a lot of information to process. I will talk more about it in the later slides. So deep into the P300, as I said, if I show you stimulus at time zero, at 300 to 600 milliseconds, there will be a wave with high peak, which is the P300 wave, and which will be which will be correlated with this stimulus. So there is how do we use? So before I tell you how we use P300, I want to see a video. See the flashes of rows and columns. That is how P300 speller works. There's no audio, by the way. Okay, so what's happening here is that uh, you get a so I told you that we have a peak at P300 at 300 milliseconds after viewing something. So when you see something that is very rare or something that is of interest to you, you will get a higher P300 peak, right? You will get a P300 peak of higher amplitude, something like this. It'll basically have a higher amplitude. So what you see here is that when when I'm showing these flashing rows and columns to the person, say I want say I'm typing. This is used for typing out letters and words. So say I want to my word starts with the letter P. Okay, so I will so this what this speller does is it will flash this column first, then this column, then this column, and then when it comes to this column, which is the column of interest to me because it has P, then you will see a P3. You will see a spike in P300 wave. So that difference, the difference is very significant that the computer catches that. And when the computer catches that, that column is selected, and then the speller goes and works towards the speller goes row wise. it will go to this row, then this row, and when it reaches this row, it will understand that I want to select P. So that is how a character is selected. Let's go over that again. Okay. So do you have any questions? We'll take a short Q&A break here.
Actually, if the person is generating, when you are showing different columns, when if the column contain P, then the
Yeah.
person's P300 the value will go up, and when you're showing different rows, if it contain P,
Hmm.
then the peak level will be reached, right? So,
Yes.
so based on this, a particular EEG graph can be obtained, right?
Right. Right.
Actually, on basis of it, if we are giving an external stimuli from outside to the brain, which will synchronize with
Hmm.
the brain signals, then we can
Hmm.
change. We can make the person's brain to develop an unfortunate emotional response when he sees a particular when he sees a particular letter.
Right. So I mean, emotions are not that simple. The when an emotion is elicited, it lights up different parts of the brain, and we as neuroscientists or cognitive scientists are still not aware of which which part of the brain is responsible for which emotion. I mean, we have localized it to a certain bit, but to elicit that, we are still not aware. I mean, you don't have to do that much to elicit a negative emotion. You can show them negative images. You don't have to do make it so complicated, right?
Okay.
Did I answer your question?
Yes, but can we control a person from outside using giving an external stimuli,
Uh,
electrical stimuli?
Not yet, not yet, and I hope not in the
Okay.
near future as well.
Okay.
Okay, so we have another question. Does this process happen quick? Yes, it is very quick. So it P300 is 300 milliseconds after the stimulus, right? So the rows move after every 600 to 700 milliseconds. The rows change. That's why you see it's happening very quickly. All right. You're welcome, Abhinav. We have seen this. Okay, so let's come to motor imagery and motor movement. For those of you who want to control neural prosthetics, pay close attention. So you see the picture on top right. This is called a homunculus. So this is the amount. This is the area of the cortical surface that is dedicated to controlling different parts of the body. So if you see here, this entire portion of your cortex is responsible for controlling just your arm, the shoulder, arm, elbow, forearm, wrist, hand, and even larger representation is for individual fingers. I mean, that is why we have huge dexterity in our fingers. I can make complex manipulations. I cannot do that with my toe fingers. Like people who write with their feet, they do that. They can do that because they create a larger representation of those fingers in the feet, right? So that is this is the reason we have huge area of the brain dedicated to controlling our wrists and fingers. All right. So whatever voluntary action that I'm doing here is controlled from the motor cortex on the brain. You see, this orange part is the primary motor cortex. This is right over here. Imagining, and even if I'm doing this, and when I'm imagining opening and closing my fist, it will generate the same neural signatures. All right. The firing pattern inside the brain is same, even for imagined movements. So it happens primarily in the primary motor cortex, which is this reddish orange part. planning of movements, like not doing the movement and planning, happens in the premotor cortex, which is the orange part of orange, light orange part over here. So and our limbs have contralateral representation, meaning my right hand is controlled by my left hemisphere, and my left hand is controlled by my right hemisphere. And I have a question for the audience. It is difficult to control limb prosthetics using EEG. Does anyone know why? You can tell me, or you can write it in the chat box. The answer is right in front of you if you analyze the slide carefully.
Yes, because imagining movement and this movement has to be imagined first and then it has to be executed. So imagining will have same neural signatures just like that of doing some activity. So if we are imagining taking a ball or if you are doing taking a ball, then both will generate same response.
Okay, so I think I have to modify the question here a bit because I think I'm not clear. It is difficult to control your feet prosthetics using EEG, feet, your leg prosthetics using EEG. Why do you think that is? I'll wait for 10 more seconds, then I will tell the answer myself. Uh, Krishna, your answer is not correct. It is it is simpler to control your feet than it is to control your hand, because your feet has less degree of freedom than your hand. Of course, there will be a delay, but that delay won't be significant, 20 to 10 to 20 milliseconds. Yes, Kriti, I think you are right, partly right, and Sinchana also is right. So yes. So okay, let me tell you the answer. So what I what we discussed about EEG, it records activity from the surface of the brain, right? And here we see the limb has representation here, which is inside, which is not on the surface, which is a little bit inside. Your hemisphere is divided into, your brain is divided into two hemispheres, and there's a huge fissure, a huge valley, if you can imagine that way, huge valley, and on the wall of the valley around here you have the representation of your feet. So when that signal reaches the EEG, it's all jumbled up, it's all converged, it's all mixed, so you can't get it, so you can't distinguish it. You'll know this is the portion, this part of the brain controls leg movement, but you won't be able to distinguish it using EEG. Okay, so moving on to steady state evoke potential. In this, the participants receive periodic stimuli. Now, don't get confused between P300, where the speed is 2 hertz, only two stimuli, 2 to 4 stimuli are presented per second. But in steady state evoke potential, the participants receive stimuli periodically at a high frequency, that is above 10 hertz. So that may be presented using flickering light. When your tube light is not working properly, it flickers. So flickering at 40 hertz will sync will work, or you can also use tones of high frequency. So I play a tone of high frequency. This will also synchronize your brain with the stimuli. When you do this, when you present flickering lights or tones, the brain activity and the stimuli start resonating with each other. You must have learned in physics what resonance means. For anyone who has forgotten, resonance is basically When a system vibrates at certain frequency, the nearby, say another system that is influenced by that system, will also start vibrating at that same frequency. So that is called resonance, and that phenomenon is seen inside the human brain as well. If I present to you a flickering light at 40 hertz, your brain activity will also resonate with it and you'll see significant 40 hertz activity. So there is an SSEP. Using SSEP, we can also do keyboard typing. So I think you need, so please pay close attention. So I have four. So I am dividing my screen into four parts. All right, and I want to know, and I tell the person to focus on something, and I will predict what part of the screen the person is looking at. And this is not, and I won't be looking at what they are looking. So what I do is I on the first block I present a flickering light, a flickering light that has a frequency of 10 hertz. On the second block 15, on the third block 20, and on the fourth block at 25 hertz. So as I said, your brain resonates with the external stimuli. So by seeing the seeing the frequency at which the brain response is coming, the predominant brain response, there'll always be other frequency component as well, because your brain has to keep working. You have to still breathe. You will see a spike in frequency of that of that stimuli. So say I'm focusing on this part of the screen which has a frequency of 20 hertz. My brain will start firing. Predominant firing in my brain will also have 20 hertz in it. So that is how I see. Um, that is how I will figure out that the person is focusing on this block. So using that, you can also do keyboard. You can present a keyboard to the person, a keyboard layout, and you flicker different components or different frequencies. And whenever they focus on something, you can basically figure out what they're looking at, what they're focusing at. All right. So what does a typical brain computer interface pipeline look like? This is what I use, and I spent 4 months figuring this out. So all the stack in this, except the EEG thing, is all the software stack is free. It's open source. Typically, if you are not good with software, you go ahead and purchase software licenses. But those licenses can go up to $1,000. But we don't want to pay that because we have open source options. So what you do is the person is wearing EEG caps, EEG electrodes. The electrodes are connected to an amplifier, because brain signal recorded at the scalp are very weak, so that needs to be amplified. Here is a computer, and the computer presents stimulus. It could be anything. It could be tones. It could be a white cross for the person to fixate on. It could also be flickering lights. There are a ton of software out there that do stimulus presentation. I use PsychoPy, which is a Python-based software. In our lab, we also have ePrime, but you have to pay $1,000 for that. There's PsychJS as well. There are a lot of softwares. So what I do is I send the EEG data on a stream. It follows a UDP protocol. It does not need to have a TCP handshake. So I use a software called EGI AMP server connector using lab streaming layer, which is again a very good piece of software. If you want to get your hands dirty with brain computer interface, you need to look at lab streaming layer. So what this essentially does is it takes the raw EEG data that is recorded in this amplifier, and it sends it in the network, all 64 channels. Whatever you decide the sampling frequency, that is number of EEG samples that are sent per second. You decide that it will send it on the network for any other process to pick it up. But along with that EEG data, I also send event markers. Right. I need to tell at this point I showed person 40 hertz flickering, and at this point of time I showed the person 20 hertz flickering. So that is also important because you have to segregate the data and then use it later for analysis. Okay, so this software along with the PsychoPy, this is PsychoPy and this is lab streaming layer, which is made by UCSD and is short Center for Computational Neuroscience at UCSD, which is University of California, San Diego in the United States. Researchers from that lab have made this software. And then when you send the data on the stream for anyone to capture, there's again I pick up that data using NeuroPipe. You can also do that with another lab streaming layer provides another software called Lab Recorder for you to record the EEG data along with the event markers. But I personally like to use NeuroPipe, which is visual pipeline builder. Basically, it takes that input data, it shows me, it plots the EEG data and tells me what is going on. I mean, you need, after you get the EEG data, you have to do a lot of pre-processing as well. So, like band pass filter, notch filtering, to remove drift, to remove the power line noise. All right, and then it after pre-processing, you can save it to save it to your disk, or you can stream the clean data again. It all depends upon your application. Or using this pipeline, you also can do further processing and you generate classification predictions, left versus right hand. If I'm moving my left hand, if I'm moving my right hand, you can also do that and you can visualize it, and you can further send those left versus right command through the network, which can be picked up by an Arduino board or a Raspberry Pi, which controls the prosthetic hand. You can purchase a prosthetic wrist online for 4,500 rupees. Uh, I am looking to purchase that to integrate it with my experiments. I haven't tested it yet. So yeah, you can send the commands again to Arduino board, which controls motors to move your hand. So this is a typical EEG pipeline. This is what I use. Many people use different pipelines as well. I'm using this mostly because it's free, and I'll be soon releasing an article on like how do you configure these pieces of software. Now moving towards the state of the art. So we know about recording the brain signals and then moving a prosthetic hand. But imagine how you would feel if you don't, if I grasp something, say this is my phone, I'm holding my phone, but I do not get the feedback through my hand. I do not get the feedback. But so what that does is that it hampers training. Like research has shown that it increases the training time if you don't give tactile feedback, if you don't give the force feedback. Now what researchers around the world are doing is that if you see here, the blue line is the blue arrow, and the blue squares here are coming to the prosthetic hand, and you see the red line, they are taking back touch commands, like how much force you are applying, or which part of your hand is in contact with the object. So these are taken and then taken back to the somatosensory cortex. Somatosensory cortex lies just behind your motor cortex over here. You see the primary somatosensory cortex. It is right next to your motor cortex, where your motor commands are generated. The somatosensory cortex gives you information about touch. So if I touch something with my fingers, that response goes to my somatosensory cortex. Right. So what this brain-computer interface does is it stimulates, or it vibrates, or it gives electrical impulses to your primary somatosensory cortex, which is your input area basically. So that input information is also given to you. So depending upon the frequency, like I told you, action potential focal frequency. Uh, so depending on the frequency, the person can figure out how much force they are applying, basically. So that is the state of the art work that's going on right now. So so as I said, we are using EEG. I'm using EEG right now. But how then in that case, if I don't want to go inside the scalp, inside the skull, then how do I give back? How do I give the commands? How do I give the touch sensation? So some labs are using vibrotactile stimulators. Basically, they're attaching vibrators to the part of your hand that is still active, and that sends commands that is still connected to your input areas. So vibrators are connected to your hand and they vibrate, indicating the force or the area of contact, and that stimulates your somatosensory cortex. That is the input cortex. Okay, so that is all for the presentation, and I will summarize what we talked about. In brain-computer interface, we bypass the normal mode of communication between the brain and the muscles. We implant electrodes, we take information, we decode it using machine learning, and then we move something. We looked at some of the common recording techniques, that is ECOG, EEG, EMG. We went over a few brain-computer interface paradigms using EEG signals. We went deeper into the EEG signals part because that is where my expertise lies. So that is where I can give you a lot of information. So we use the P300, we use P300, we use motor imagery, motor execution, and we use the SSVEP. And we also saw what a typical brain-computer interface pipeline looks like, which is right over here. Uh, Thank you for your attention. Any questions? You can note down my email address if you have any follow-up questions that you don't have any at the top of your head.
So if there are any more questions, you can ask him directly or you can use the chat box.
Actually, premotor cortex will be just some idea that will that ideas will be going to primary motor cortex, right? So if there are two.
Your voice is breaking. I can't understand what you're saying. Maybe you can type on the chat box. How do we? So we have a question. How do we make behavioral inferences from EEG data? So as I said earlier, the EEG data in itself is like white noise that you see on your old CRT TVs. We can't make out much from it, but we can certainly see the differences between behavior. So if I do one action again, I'll talk with respect to hand movement. Say if I move my left hand and I move my right hand, if I compare the signals, you will definitely find some differences. Behavioral inferences are more difficult to make because there has to be some incremental differences, right? So there's a classic experiment that I would like to talk about. So I'm telling a person when they see something, when they see an X on the screen, like a person is repeatedly shown stimulus, shown characters on the screen. And if I tell them that when you see an X, you press a button, and the reaction and you see something, you see a typical brain response, you see a brain response. All right. Now I tell the person that if you see an X, you press the left control button. If you see a Y, you press the right control button. Now, if you compare a brain activity from those two task conditions, whatever difference you see will be the decision making aspect. Should I press the left button or should I press the right button? So in that case, your frontal cortex will light up heavily because that's a decision making task under pressure. So that's how we make behavioral inferences. We do small changes in behavior and then we see the differences. I hope I answered your question.
Yes. Yes. There will be two, and that will be stored in premotor cortex, and based on this, yes, response will be there. Yes, sir.
Yes. Okay, so we have another question. Rather than installing a device directly to brain, is there any way to directly influence a single or a group of nerves of particular region? Yes. We also have TMS. It's a system where you can. It's a huge metallic block which has magnetic coils inside. So what it does is it stimulates a part of your brain by giving it electrical impulses, magnetic impulses. That is called transcranial magnetic stimulation. So using magnetic fields with, from two coils. I don't know. I exactly don't know the physics and electronics behind it. But yes, you can influence a group of nerves. For influencing a single nerve, you have to go inside because these are very small cells. Welcome.
So using your brain, can we able to control the other person's action using this method?
Uh, that will have several implications. Like you will need ethical. That has ethical implications. You need that person's consent. Theoretically, yes, you can do that. You
Thank you,
may
sir.
be able to control. I wouldn't advise doing that. What are the requirements to build a web application with help of EEG headset? Uh, Wait, I am not sure about the question. Web, can you give a few more details as to what this web application does?
Yeah, actually, as of now, I saw like EEG things are more into robotics and medical field. So suppose
Mhm.
we want to bring it on the entertainment field, or if suppose I want to create a web application where I can design or something with the thoughts. So I know it's like very complex thing, but
Mhm.
is it any possible for developing
Well,
a
I won't.
soft web
Yeah,
application?
I mean, I talked about brain typing with your brain. If you're talking about coding the web application, then I think the person will take few days to just to type "Hello World." But
Okay.
designing, I'm not sure how that will work because aesthetics and design are not my area of expertise.
Okay.
I mean, you're free to look into it. I'm sure people are doing work on that. Go to Google Scholar and type your queries. It'll give you something definitely.
Sure. Thank you.
Welcome.
So I hope there are no more doubts. So this session was definitely worth the time. I'm sure that everyone who attended this session learned something new. I'd like to genuinely thank our speaker for the day, Shivam Chowdhury sir, for sharing your knowledge with us. I thank
Thank you, Aishwarya.
everyone
It
for
was
attending this
very.
session.
I am very happy that people interacted and asked questions. If you're generally interested in this field, I would urge you to, I mean, get started with YouTube videos. There are a lot of inspiration out there, and then you go into neuroscience. Thank you all for coming.
Thank you, sir. The recorded video of this session will be posted on our website. I have posted the link of website in the chat session. And also, the link for feedback form is also posted. Make sure that everyone would leave a feedback. Once again, thanking everyone on behalf of EETI Foundation. Thank you.